Hello and welcome. Thank you for attending today's session. My name is Aaron Hobner, and I'm with Solarity Incorporated, a Columbus, Ohio-based manufacturer of advanced cell culture products. We've been making the Petaka G3 since 2009, so we have a long history, and we've developed a number of unique methods and applications for your cell culture that I think you're going to find very interesting. By the end of the talk, I hope to introduce you to some really novel concepts and some new applications that you can use to improve your cell culture. So on the right, you'll see the basic specifications for the Petaka G3. It's a single-use sterile cell culture device. It's made of Class 6 medical grade polystyrene resin. It's optically clear and it has low autofluorescence. You'll notice it's a very slim, compact design. It's very different than your standard cell culture ware. It's functionally closed, and the main culture chamber fills with up to 24 mils of filling volume if you're growing suspension cells. And if you're growing adherent cells, each Petaka delivers up to 150 square centimeters of cell culture surface. So you can cultivate a lot of cells in a very small space. We're going to show you how. The main feature of the Petaka and why it has the unique design is because of the core technology that controls dissolved gases, including your culture, the dissolved oxygen in your culture, and the CO2, which of course regulates your pH. This leads to a lot of unique, very useful and interesting applications. And it's going to be the main part of the talk today. So I'm going to describe things like live cell shipping, for up to 10, even 14 days to get rid of some of your cryo processes. I'm going to show you how you can centrifuge directly in the Petaka to pellet and wash to get rid of unnecessary cell transfers. And it's functionally closed, which allows a lot of applications that you can't get in, let's say, flasks or dishes. Things like benchtop monitoring, long-term microscopy without any hardware. Plus, there's some inherent design features, like the fact that the Petaka is individually bagged, has a permanent UDI label that's barcode readable, that I think is really good for sterile applications, GMP and cell production applications that you just can't get with standard cell culture ware today. Any type of cell that you can imagine has been successfully cultivated in the Petaka to date. So primary cells and immortal cells, patient biopsies, every kind of stem cell imaginable. We have protocols and we have experience with all of this, so we're confident that no matter what you're cultivating, it's going to do very, very well in the Petaka. And in fact, the cells are generally going to feel more at home in the Petaka because, like I said, we provide that in vivo tissue-like culture environment that you can't really get with any other technology. So just so you can become familiar with what it looks like and how it works, here's a typical cell culture setup. I want you to notice from the beginning that I'm not reaching into bags and contaminating flasks and dishes and rewrapping them as I bring them out into the laminar flow. Each Petaka is individually bagged sterile, so it goes into the laminar flow sterile, and there's never any risk for contamination. You also notice they stack in very tightly, so I don't have stacks of dishes and plates that I have to fill, taking up all the workspace. I mentioned all the liquid handling goes through the sterile resealable silicone port. Because of this, we recommend using syringe-based transfer and blunt cannula, even though pretty much every method of liquid transfer is still possible with the Petaka, and I'll show you how that works later. This is a very common way to fill. Most people pour their media into like a 50 ml conical, for instance, and pipette it into their dish or flask. Well, instead of using a serological, we simply use a more closed hygienic system like the closed syringe. So we aspirate out of that. We thought this would be a good demonstration. Here's the media going into a Petaka. Notice everything goes in using a blunt cannula for safety, goes in through the resealable silicone port. Filling goes from the bottom to the top because it fills through the channel on the right side of the Petaka. and you just fill it right up to the top. The only trick is you don't want the media to go into the microcapillary chamber at the top. So if you just fill the culture chamber, you have a perfect fill. Notice it was relatively quick and easy. And I want you to also notice how hygienic it is. So once you aspirate all the media up, delivery is very quick, very hygienic. It's a functionally closed delivery. So you're always going in and out of that sterile silicone port that can be re-sterilized whenever you want. People cultivating in 96 well plates also are familiar with aspirating out of these sterile basins. This is also very convenient, particularly for large volumes. If you're doing repeated fills and you want to fill 6 or 8 or 10 Pataka, the basins are really, really useful. Okay, so I also hope you notice that there's no particular training required here, no special motions. It's very simple. The workflow is easy. I think anyone who's been doing cell culture for even a little bit and has handled a syringe before can get used to this, in minutes and master it within days. So it's very simple. Like I said, the sterile silicone port accepts all kinds of tips. So whether it's a serological with an adapter tip, or it's an Eppendorf style tip, or repeater pipettes, everything's possible to move liquid in and out of the Pataka because the silicone port's very, very flexible and adaptable. So once you have your Pataka filled and you want to take it to the incubator, we don't have you just stack it in loosely. We provide a silicone stand system for the Pataka, so you can easily keep them compact and transport them to the incubator. In this case, I'm going to show you how that's done. I have my Pataka ready on the left, my incubator's on the right. Transport's easy, it's compact, it's secure because every Pataka is locked into its slot in the stands. And for suspension and adherent cells, they go in horizontally in the beginning. And if it's an attached cell, you'll see you want to leave them horizontal so they attach. And then once they're attached, you can actually turn the stand vertically. And that does two things. It saves footprint space, and any cellular debris goes to the bottom and stays off your cells. It's a much better way to cultivate adherent cells. This is to demonstrate the cell density you can get in the Pataka. So if you fill the stand with 12 Patakas, that space right there is the equivalent of 24 T75 flasks. Finally, another process that's unique to the Pataka is direct centrifugation. We don't centrifuge in the standard 15 and 50 ml conical tubes. We pellet and wash directly in the Pataka. We think it's a much more hygienic way of processing cells and reduces contamination risk. So most labs are growing their cells in an extremely hyperoxic state, typically somewhere between 3 and 7 times the dissolved oxygen that the cells experience in their native tissue. The literature is pretty clear that this impacts practically every major aspect of cell physiology, from growth and apoptosis, cellular senescence, the protein expression profile, and it also leads to changes in morphology, migration, and significant changes in epigenetic markers. The idea behind the Pataka was to get this under control and deliver true physiologic and actual physiologic tissue levels of dissolved oxygen, but without the tri-gas incubators, the glove boxes, and the end-to-end cabinet systems that are typically required to deliver lower physiologic oxygen. The only difference is the Pataka system does it in a way that's passive, and it's automatic, and it's controlled by the cell, and it's adaptive to the cell's needs in a way that these programmed atmosphere control systems really can't deliver. So we think we have a better solution, not only from a cost and a workflow perspective, but also we think we just deliver and adapt to the cell's needs much better, because we have a responsive system, and it's all based on the microcapillary system in the Petaka, which I'm going to show you in the next slide. Every Petaka features a microcapillary gas control system which prevents the unrestricted diffusion of gases into and out of your culture. So as the cells metabolize and reduce the dissolved oxygen in the medium, the Petaka adapts and increases the diffusion to meet that demand. Now, this results in an equilibrium being established that matches the dissolved oxygen of native tissues, and this occurs within only hours. And because the system is automatic and passive, that equilibrium is maintained for the life of the culture, typically out a few days. Now, just as the Petaka regulates the dissolved oxygen, it likewise restricts the diffusion of excess CO2 out. So it creates a local concentration such that your culture sets up an equilibrium equal to about 5 or 6% of atmospheric CO2 that you currently enjoy with your current incubator. So this all results in the only device that delivers automatic, self-regulating, physiologic gas control, and it does it at the pericellular level. And there are a number of systems out there that purport to do something similar, but we think we have an advantage, and we think we deliver a much better in vivo-like culture experience because we're not attempting to control the atmosphere. We actually deliver exactly the amount of gases that the cells need, no more and no less. And we don't try to set an artificial level on any different cell type, which can vary from line to line and culture to culture. So we think we have really what amounts to the perfect system to deliver that native tissue environment in an in vitro setting. Because the Petaka is functionally closed and provides excellent control of dissolved gases like CO2 and oxygen, you can do things with the Petaka that you can't do with open systems outside of the incubator. So we make the Petaka DO sense with an oxygen sensing dot, and that's matched with a non-invasive light-based reader so you can get real-time dissolved oxygen. Now this is a great feature because it's off the shelf and ready to go this way. So whether you want to follow the dissolved oxygen in say for bioprocess monitoring, or you want to do some sophisticated experiments of dissolved oxygen for metabolism, this can all be done in a very easy way now. So for example, you can have many cultures growing in the incubator in Petaka, and you could take one representative example for the lot and do a bioprocess monitoring. And not only is it convenient to do the dissolved oxygen monitoring over many days on the open bench top, because the Petaka features the resealable port, you can take many small volume samples and measure different analytes like lactate and pH and amino acids. So this opens up a new paradigm for really easy, low-cost bioprocess monitoring and monitoring for your experiments during scale-up that you really can't get with any other system, or at least you can't replicate in a convenient and economical way. And this also shows the extreme flexibility of this system because being able to get out of the humidified CO2 atmosphere enables a lot of very easy process workflow that we think is impossible with any other technology, and we're very proud of that. And here's just some examples of some of the experimental profiles that we've recorded where you can match cell growth. So we'll take a suspended cell line and we'll take samples over many hours and many days, and we can get cell counts, we can get pH, we can get lactate, glucose, and then compare that over time with the oxygen level, and then take the first derivative and look at oxygen consumption rate. And we can do these really sophisticated and wonderful experiments, really important for cell metabolism, but also, like I said, also applicable for scale up and bioprocess monitoring. And again, this ability to multi-sample while getting cell counts and oxygen, pericellular dissolved oxygen, you really can't do that as conveniently with any other system without interrupting the oxygen control or going into the incubator. And so we think we have probably the most versatile system in the world for this kind of monitoring. I alluded to cell production scale up earlier, but I want to take some time on this slide to highlight the benefits of scaling up in the Petaka in the stand system. So on the left, you have the Petaka stand that can hold up to 12 Petaka, which is the equivalent of 24 T75 flasks. And if you compare that to the flask on the right, you can see how we easily get 20 times the cell density for the space and the footprint that you get if you were sticking with flasks. So one Petaka stand easily produces 250 to 300 million cells. In fact, I can easily produce 1 billion cells on one shelf on my small incubator. That's not a problem. In terms of all other ways to scale up, I mean, there are these stirred tanks with microcarriers, and there are these mega multi-tier flask systems out there, and there are other systems, but they necessitate changes in protocols. So you have a change control there. You have to revalidate. There's capital equipment costs with many of them, and we think we hit the sweet spot because you get to scale up using exactly the same methods and protocols from your early work, and whether you're moving into animals or a pilot scale or an early trial, you get to keep the continuity of your method from beginning to end, and you get to scale up. You don't have to have any big jumps where the capital equipment systems would going to necessitate the big jumps, and you may not want that. With the Petaka, you can always just add a Petaka and add in 15 to 20 million cell increments. And if you want to add 300 or 500 million cells, you just add 1 or 2 stands. And of course, harvesting is always very easy, very clean. Everything goes through the sterile silicone port. So we have a lot of laboratories that are using it in a GMP compliant way, and it's all available. So we think we have really one of the best systems for that medium volume scale up, and particularly for the early pilot work, and like I said, the translational scale up work that's being done out there. So this is something we encourage you to check out as an alternative to the more expensive and complex systems out there on the market. There are a number of new and extraordinary applications made possible by the Petaka. One of them is ambient temperature cell storage and transport. There's a natural phenomenon called cell pausing, where when you have low physiologic oxygen and mild hypothermia about room temperature, the cells naturally go quiescent, and they basically hibernate. Metabolism's dropped 80 to 90%, and it's an excellent way to temporarily store cells and prepare them, for instance, for process storage or shipping. And the Petaka exploits this natural process by providing all the protections at the low oxygen, while also managing and balancing the pH in a container that eliminates dehydration over short periods. In doing so, the Petaka makes available an alternative to cryopreservation that's never been practical until this time. Ambient temperature cell preservation in the Petaka is now being carried out by many labs all over the world, including academic labs, cell therapy manufacturers, and GMP cell processors. It's a very mild and natural effect. The cells come out with no genomic effects, no mitochondrial defects. It's just a gentle process, and you get a very, very good result. On the left, you're going to see a chart that displays the cell viability at room temperature preservation in the Petaka at 7, 14, and 21 days. This is really representative of the more than, say, 50 cell types that Celardi has tested in this paradigm, and the results are all very similar. That's why we say 7 to 10 days for almost any cell type. You're going to get very good viability, 90+%. Of course, we encourage you to test and validate this in your own system. But if you're looking to smooth your process window, if you're looking for a little extra time so you don't have to come in and do processes on weekends, if you want to ship cells to colleagues, or if you have a commercial product that you're just looking for an alternative to cryopreservation and cold chain logistics, this is something that's robust. It's been used across the world for many years, and it's a universal application that I think you might want to investigate. So the Petaka is very good for all of the standard transfection modalities, whether it's viral or lipofectamine. Your results are going to be very similar to what you'd get in the standard labware. But the Petaka does some things different. It's been validated on the Sonicure system as the cartridge for the focused ultrasound-based transfection. We encourage you to reach out to that group. Also, the Petaka has been validated by OZ Bioscience and all of their magnetic-based transfection protocols. So with the Polymag system, it's very easy to, for instance, transfect 2 cells on either side of the Petaka wall with one vector, or you can even do like a co-culture system highlighted in the bottom right, where you might have cell type A and B with 2 different types of transfection products. And this is presented here. We actually kind of took what's possible to the extreme and said, you know what, we're gonna seed one wall of the Petaka with COS cells, the other with HEK, and then we're going to do a dual transfection. So the one gets a GFP and the one gets an RFP, and you can see just using the magnetic-based transfection, we get really good clean results, high efficiencies. And this is something that's really not easily replicated, even using the hanging well type systems. You get a large number of cells in co-culture, 3 millimeters apart, in direct liquid communication with each other, each expressing and being managed and manipulated in a very controlled way. So we think the Petaka opens up possibilities because it has a different design, and it's amenable to all of these the methods that are available on the market. So if you're doing this kind of work with co-culture and multiple expression systems, you might want to check out the Petaka for these applications. We have white papers and notes, and again, all of these protocols have been tested and developed, and we can consult with you on setting up any specialty experiment. If it's something that you're considering, we may have already even done it or had a customer that's done something similar, and we think we could give you some good advice in that regard. So the Petaka has unique applications in cryopreservation, and I want to take a couple slides to talk about this because it is something new and really special. So I'm not going to go too much into the problems with the current methods for cryopreservation using the cryotubes, but basically you get a lot of trypsinization injury and DMSO toxicity, and then there's the slow freezing and slow thawing with the water bath. And it's just a lot of injury to the cells, and there's a lot of transfers. And of course, it's expensive to ship. And I just talked about the alternatives to cryo, but now I want to show a cryo alternative in the Petaka that eliminates all of the transfers in the cryotubes. And I think for anyone storing away large amounts of cells, or certainly clinical samples where you don't do all the sub-aliquots, I think this is something that could be really interesting because, again, it's totally new. So I'd like to discuss the concept of direct cryopreservation in this primary cell culture container. So there's a method called thin film cryopreservation, and it's very well researched, and there's a lot of reports in the literature. And there are tremendous advantages to this, particularly if you have a concept where you want to freeze away cells in a monolayer or a sheet for a regenerative medicine application. Freezing the cells attached leaves them in a native configuration. You have cell-to-cell contact. They're on their native extracellular matrix. Plus, compared to cryotube freezing, you're not trypsinizing and you don't have to leave a lot of DMSO. Well, the method is relatively simple. You can do a brief exposure to a cryopreservation media with, let's say, typically 10% DMSO. But instead of leaving a large volume, you can aspirate everything off, and that's particularly easy in the Petaka. And you just leave a thin film, maybe 10 microns, and then when you put the Petaka into the vapor phase or into an ultra-low temperature freezer, you get very rapid freezing and even vitrification. So the cells are protected extraordinarily well. And then, of course, the freezing's a breeze. Just all you have to do is fill the Petaka with warm media, and you can fill directly with the warm media. You don't have to do the external water bath. Now, This is not practical with dishes or plates or flasks. Even some bag systems are rather bulky, but with the Petaka you can get, of course, such a cell density as compete with a typical 9x9 frame of 1 mil cryotubes. In a cryo box, you get approximately the same number of cells if you're freezing on both sides of the Petaka. So not only is it easy, it's also practical, and you can freeze away cells in a number very comparable, let's say, to boxes filled with 1 mil cryotubes. And so for anyone biobanking or storing away cells who might want to ship assay-ready cells, where the end user only has to add a little media and they're up and running right away, or you want to do long-term cryopreservation, or you're thinking of a way to cryopreserve a sheet and keep it intact. Like I said, it's a lot of research, great advantages, a big upside, but until now, there's never been a commercially available product that allows this in a practical way. And now it's a possibility. So we invite you to check out this application too. Again, another first from the Petaka. Since the Petaka maintains the in vivo culture environment independent of the incubator, it makes some new applications possible. And one of those is a twist on, say, long-term microscopy. So if you're doing any of this work and you want to take long-term images, this is a really good alternative that doesn't require any special equipment around your microscope stand, and it doesn't require any special microscopy equipment that can go into the humidified space. Here you can see we seed cells in the Petaka, and all we do is provide any one of a number of commercially available warming films, and we maintain 37 degrees. And it's very easy and low cost way to move into some good high resolution long-term microscopy for practically no additional cost up front. We think this is a great alternative, and it further highlights the novel applications that are possible with the Petaka. Finally, direct centrifugation is possible in the Petaka. This is good for a number of reasons. One, you're not transferring and you're keeping your cells and all of the whole process, whether you're concentrating or washing, separating cells, you're doing the process in this primary sterile container. We think it's superior to the old method where you have to move everything in and out of the hood in 15 or 50 mil conicals. We do all of our pelleting directly in the Petaka. We can wash, we can rinse, we can dilute. We just think it's a better process. So particularly for anyone who's worried about contamination or has really critical samples at some point, or has experienced some contaminations before with all the transfers and movements, we encourage you to check out this method, particularly for GMP processes where the Petaka offers distinct advantages like individual bagging to move between clean rooms, and where you want to streamline your workflow and really reduce your contamination points. We think this is a great alternative, and there's really nothing else on the market like it that we know of. In a way, this slide is like a summary of the whole talk, where we've been comparing the unique advantages and the features and the benefits of the Petaka and all the methods and applications that it allows, and comparing it to traditional cell culture ware. And we summed it up that, the problems with the traditional products are they're bulky, they take up a lot of space. Everything involves an open transfer, for instance, like the centrifugation, which opens you up to contamination. Classic culture is hyperoxic. The disposables provide no pH control. You're stuck to the incubator because you need the humidity and the CO2. You can't centrifuge directly. There's no chance of cryopreserving directly, except a little bit in some bag systems. And you can't ship cells live, so there's no alternative to cryo if that's something you're looking for. And again, the workflow is just way more complicated and wasteful, even in terms of plasticware. And I want to show you a little chart to highlight this. So in this, we tried to combine a few measures all in one. So on the top, you see a typical workflow, and we imagined getting a cryotube out of the dewar, putting it in the primary plate, culture expanding it, and then splitting it into daughter plates. and then taking one of those, the cells out of one of those daughter plates, and then preparing it for shipping to a colleague or a clinical site, for instance. And you're moving from, if you look at the black dots, that's the classic method. You're moving from cryotubes and you're transferring with serologicals into conical tubes and centrifuging to wash out the DMSO and concentrate the cells and split them. And of course, you're releasing with a scraper or a trypsin at every step. And then once the daughter cells are ready to go, you still have to then do a cryopreservation protocol. You're spinning it down, you're adding cryoprotectant, you're putting it in the dewar, and you're shipping on dry ice. And then in the final use, your recipient has to then thaw it out, rinse out the DMSO, and prepare the cells for final use. Now, that exact same procedure can be performed in basically 2 Pataka with only 1 centrifugation step and no cryopreservation. So we imagined you thaw out a cryopreserved Pataka simply by adding some media. And then when it grows up, you do 1 split. So from a parent Pataka into the daughter Patakas. And then once they're confluent, you simply put it in a shipping container, use a shipping protocol, ship live, and then the recipient gets the cells alive, healthy, and with no downstream processing required. And we think, without overstating any of the processes at all, you can look at cell transfers, and with the flask-based system, and this goes for, of course, dishes and plates as well, you're looking at 8 cell transfers in 7 containers. Well, with the Pataka, you're looking at 1 transfer and only 2 containers used. The parent Pataka that came out of the cryo, and then the daughter that was grown up and then shipped to the end user. And you only have 1 centrifugation step. And we think this is a nice illustration of how we streamline and make more efficient the workflow. But not only that, your typical process, we've noted and we've tracked this, you can eliminate probably 70 to 80% of your plastic waste by switching to the more advanced system. It's much more efficient, it's a lot cleaner, and again, your workflow is smoothed and minimized such that you just don't generate the waste in not only product, but in time and all the steps that you get using the classic method. So we think the Pataka beneficially reinvents your classic workflow and allows for reductions and improvements in efficiency that you just cannot get with any other system that we're aware of. And we invite you to talk to us about this and let us know what your pain points are, because we have seen this transformation in many labs where things just get tremendously better when you move to a system that encourages efficiency and waste reduction. And so finally, most every talk ends with some kind of summary slide, and we have a chart here. And on the left, we have all the features that I went through, oxygen and CO2 regulation and humidity control, and all of the special advanced and novel applications made possible by the Pataka. And we compared it to a lot of the standard labware, including Petri dishes and roller bottles and bags and flasks. And we don't think it's a stretch to say that the Pataka excels at every one of these special applications and provides all of these special advantages, whereas very few of these features and methods are even possible in any other cell culture product. And even then, when it is, there are workflow improvements with the Pataka that this chart doesn't communicate. But we would be happy to communicate that with you in a session if you want to explore any of this. So to summarize, we just think the Pataka is a better system, and it really doesn't matter what you're doing, whether it's basic research or you have an applied process, or you're in a clinical trial, or you're handling patient samples. There's almost always some process that can be improved when you introduce the technology that the Pataka gives you, because it's a technology that provides tremendous new capabilities, and it just has to be explored and tested. But we think most labs can benefit from the Pataka in one way or another, and we'd be happy to show you how that could be. So you can reach out to Solardia directly for product information and support. But we also have an excellent network of distributors worldwide. So if you recognize any of these names or you're working with them already, we encourage you to look for the Petaka product through your regional distributor. Also, please check out the Solardia website. We have really great write-ups in our Solardia blog section on really all of the applications that we describe here. We go into depth, we present a lot more detail and a lot more data, and we get into the theory and the ideas behind how these systems work and ways to make all of these processes better. You get a lot more detail in these write-ups. And then, of course, you're already on our mailing list, so you'll probably get updates on upcoming webinars. And you can imagine that any of these applications can constitute their own webinar because they're new and exciting and unique to the Petaka. So look forward to further updates on some of the upcoming webinars. We're very excited to deliver these to you, not only because we think it's valuable information, but again, because so much of these are only possible in the Petaka, and for us, it reminds us how special this product is, and we're just happy to share that with any of our customers. So thank you very much again for attending. It was my pleasure to share this session with all of you, and we look forward to speaking with you soon. Thanks.